All right, hi there again. This is going to be Chapter 12. This is 2401, Chapter 12, which is nervous tissue. So this is kind of introductory to nervous tissue. This is, by the way, the first chapter of the last unit. So this will not be covered on Test 3. This will be covered on Test 4 and the final. So let's get started. So first off, just an overview of the nervous system. You have two basic divisions, I suppose. The central nervous system, which is your brain, and your spinal cord runs right down the middle. The central nervous system does most of your processing. It does most of your sending of impulses out to the body. And obviously we will get to the brain and spinal cord in detail later. The peripheral nervous system, peripheral means off to the side, so peripheral nervous system is lateral to the central nervous system. Primarily this is cranial and spinal nerves. You're learning in lab about cranial nerves where they kind of emerge from the underside of the brain, and the spinal nerves come out from the sides of the spinal cord. And there are branches as they get farther out into the body. There's two subdivisions of the peripheral nervous system. The afferent or sensory division. Afferent means towards, so afferent is going towards your central nervous system, and it's sensory, that's kind of the function, in that when I whack my hand with that, the impulses are being sent up to my spinal cord and to my brain so I can process that and wonder why am I hitting myself. The other division is efferent or motor. Efferent means away from, so the efferent system allows me to catch this thing like this by sending out impulses to my muscles. That's why they call it motor. Sending out impulses to my muscles, which then can act upon those impulses. Now, there are two subdivisions of that efferent system, and these are, you've heard of these before, sympathetic and parasympathetic divisions. The sympathetic division is oftentimes simplified as fight or flight. So this is going to be activities that require immediate, high energy reaction. So if you're going to play in a sporting contest, or you have to fight a bear, that's the one I always use. So if a bear runs out from behind the tree, you better go into that. You better engage that sympathetic division because you're going to have to do something. What it involves is your respiration goes up, your heart rate goes up, your blood pressure goes up, your pupils dilate. You basically get ready to go. Parasympathetic division is sometimes rest, maybe rest and digest is a good way to say that, and that's kind of lower energy activities. You do this when you're sleeping. This is the division that dominates when you're sleeping, and when you're eating, or when you've just eaten and then you fall asleep. All right, so let's look at the real nuts and bolts of the nervous system, or of nervous tissue, and those are cells called neurons. So a neuron is different than a nerve. Keep in mind, if you hear the term nerve cell, they're going to be talking about a neuron. If you hear the term neuron, I'm talking about a neuron. If you hear the term nerve, that's talking about a large multi-neuron cable that runs through your body. You can think of those in a similar way as muscle fiber or muscle cell versus muscle. So neuron is a cell, nerve is a bunch of cells. And I'll discuss those later. Some key things about neurons: they live a long time. This is a good thing for us because our brain is made of neurons, and they all have these neural pathways that you send impulses around. This is how you keep all your memories. This is how you accrue wisdom. You gain knowledge. And if you had the neurons dying off all the time and being replaced, those neural networks wouldn't remain the same, and you'd have big gaps in your memory as well as other stuff. So they live a very long time. They're amitotic. That's mitotic. That's not an N. It's T-I-C. So when you get to be an adult, you're basically not making any more neurons. You're not making any more of these cells because you've gotten to your full human size, and those neurons which you've set up now have to build upon all of the neural networks that they've already established. This is also why when little kids are developing from year to year to year, their personalities and behaviors and what they do changes dramatically because their nervous systems are constantly morphing. Once you get to be an adult, 20s or so, you basically kind of settle in on what you got. They're very aerobic. They have very high metabolic rates. They don't do well if you deprive them of oxygen. They don't work well anaerobically, unlike your muscle cells, for instance. this is why you can get brain death really quickly if you get oxygen deprivation to the brain. There's a couple of parts here I've listed, and I'll show them up on the board here in a minute. There is the cell body, which is just the main, mass of the cell. If I was going to draw one, I'd do something like this, where I'd go like. So the cell body is basically this thing right here, and it's got a plasma membrane just like any other cell does. These little extensions off of that cell body, and I'm just drawing one example of a type of a neuron. These little extensions right here are called dendrites. I've said that they're both receptive, which means that both the dendrites and the cell body are going to be taking in, they're going to be receiving messages from the outside. This is where other neurons send their axons to send impulses, send messages to this given neuron. So they're receptive. They are generally clustered in your central nervous system. So you find most of your neuron cell bodies in your brain and your spinal cord. They call the pockets of these, they call them nuclei. So the term nucleus is used for a lot of things in science. The center of an atom, the big organelle in a cell, but it's also a cluster of cell bodies in your central nervous system. So that adds to the confusion in the use of that word. You do have some in your peripheral nervous system, primarily involving sensory neurons. When they come back into your spinal cord, this will be something that you'll cover in depth in lab, and I'll point out next week, I think, or the week after, or next lecture, or the lecture after about this. But these clusters of cell bodies in your peripheral nervous system are called ganglia. I'm not going to draw the spinal cord transverse section, but you'll see it later. As I mentioned, dendrites are receptive. You can have many dendrites per cell. You can have dozens more than that. But you're only going to get one axon. So there's going to be one axon leaving that cell body. And this is the transmittal part of the neuron. This is going to be the part where once this cell body basically decides to send an impulse, it's going to send that impulse down this axon. Then you've got some other structures out here. These little bulbs here I'm putting at the end. If you remember from the muscular system, I had something called an axon terminal. Well, that's the end of one of the branches of an axon. So when this neuron sends an impulse, that impulse travels down the axon, spreads out to all those different branches, goes to all those axon terminals, and everybody at the end of those axon terminals then is going to get that same message. They're going to be receiving some neurotransmitter that axon or that axon terminal will produce. All right, so let's look over here. Let's look up at the board here at the projector screen. And I want to point out some neuron structural types. Are we center screened? Okay. So you can see that there's 4 basic types, and really three that are super commonly referenced. And of these 3, really only these 2 are really the main ones that we're going to be talking about. This is the big one over here. This is the one that is the most common. 99% plus of your neurons are this structural type, and these are called multipolar. They're called multipolar because there's one cell body and 1, 2, 3, 4, 5 poles, many processes that come off of that cell body. Now, if I ask you about the structural classification of a neuron that looks like this, or a neuron that has a certain job, you'd say multipolar. So structural classification is how it's built. Functionally, multipolar neurons, and I don't have this written down anywhere, and they don't have it here either, but functionally they act as either motor neurons. So when I send an impulse to my hands to grab this stick, the neurons that take that message to my arm muscles are multipolar neurons. They're functionally motor neurons, and then they are built structurally as multipolar. But multipolar neurons also serve another job, and that is to process all of this stuff in your brain. So the vast majority of all of the nervous tissue in your brain and your spinal cord is the nervous tissue of multipolar neurons. When they're in your brain or in your spinal cord and they're sending impulses back and forth between them, they call them interneurons. Now you may see the term association neuron, but interneuron is the word that I generally use. So write those down. You're gonna have to write down these functional terms. So multipolar neurons either act as motor neurons or interneurons. Motor neurons go out to muscles, or glands, interneurons do all that processing. So when you're thinking about what to write down right now, you're thinking about this when you go, what did he say? That process is being carried out by interneurons, which are multipolar in structure. Let's go over these guys. Unipolar may look like there's two poles, right? But really there's only one process coming off of this cell body. If there's one process, you call it a unipolar. These are pretty much exclusively sensory. So when I touch my hand with this stick, I can, without having to look at it, I can tell I'm doing it, right? I can feel it using my sense of touch, and that impulse is being sent up my arm to my spinal cord, and then that impulse is transmitted onto another neuron, the interneurons in my spinal cord, which then I'm going to have to do something about it, right? But that impulse then sent up a unipolar neuron, and that's why, and then functionally you call that sensory. So unipolar structural shape, sensory is the function. Over here we have bipolar, 1, 2 poles. These are also sensory, but they're very limited in their scope. You find them in your retina and in your olfactory tissue. So these are sensory, but they're really specialized as where you find them. We'll talk about those in the last chapter of lecture. Lastly, this one's called anaxonic. Anaxonic. So an is without in this case, without axons. And these are all interneurons. These are not very common. You find them in the central nervous system. Now let's look at this next little image right here. And this shows kind of two, I guess these would be probably two interneurons, or it could be an interneuron and a motor neuron, whatever. But it shows two multipolar neurons interacting. Over here on the left, we see the neuron that's sending the impulse, and you can see this little arrow here. I'll talk more about that later, but it's sending the impulse down the axon. When it gets to the end of this axon, it gets to these branches, you're going to have the neurotransmitter released at these little gaps, which you remember that term synapse, that's a little space, that's just all that means. So you have the neurotransmitter released at a synapse, which will then signal this neuron to do something or potentially to do nothing. Sometimes you can signal a neuron and its response is to not do something, which I'll get into. A couple of things you might want to notice here before I move on. Let me see what my next slide is. Yeah, a couple of things you might want to notice on this slide before I move on to the whiteboard again is that you see these little hot dog buns or whatever it looks like that are wrapped around this axon. Well, these little hot dog buns, they call a myelin sheath, and I'll break it down. There's a cell called a Schwann cell, which I'll get into, that makes that substance called myelin. And what this does is that it's going to dramatically increase impulse transmission speed. So you're going to go from a very slow wave-like transmittal of the impulse to a really fast one. And I'll describe the function of that. I'll describe how that works later, the mechanics of that. You also see presynaptic cell and postsynaptic cell. So if the synapse is right here at these points of near contact between cell A and cell B, this one is, the one on the left is sending the signal, so it's before the synapse. This one's receiving the signal, so it's after the synapse. And you see some of the other structures labeled that I've already described. So let's now move over to the board again, the whiteboard. And got some more terminology here. You see I've got synapses up there. Synapses consist of a couple of parts, as I mentioned. The presynaptic neuron, which I've described moments ago. You may see it called the presynaptic membrane. And so same thing. If they say presynaptic membrane, they just mean the membrane on the presynaptic neuron. There's a synapse or synaptic cleft. I tend to use those things interchangeably. So whatever. If I say synapse, I simply mean the gap in between. And then the postsynaptic neuron, or you may see postsynaptic membrane. Now, neurotransmitter is what's produced by the presynaptic neuron. So I'll draw that in right here. Here comes my axon with a big bulbous axon terminal at the end. And then I'll draw like maybe here. That's the membrane of the postsynaptic neuron. Well, what this presynaptic neuron does is that once it transmits an action potential, which I'll refresh your memory on here in a bit, once it transmits an action potential down to this axon terminal, you're going to get an influx of calcium. Remember, calcium played a part in muscle function as well. Calcium is going to flow into the cell, which is going to induce the formation of these vesicles. Those vesicles contain a substance generically called neurotransmitter. So neurotransmitter, our most popular model of neurotransmitter is acetylcholine. I thought I'd written it down somewhere. Where did I write it? That's just one I did. Acetylcholine. I'll find it later. Did you see it, Brian?
I thought you wrote it down too.
Me too. I thought I wrote it down too. Anyway, acetylcholine, which I will write. I promise I'll write it down later, but it's acetylcholine, often abbreviated just ACH. So, a neurotransmitter, primarily acetylcholine, has been formed in this axon terminal, and you're going to release it through a nice simple exocytosis. And if you remember exocytosis, it's just this big vesicle meets the edge of the membrane, pops kind of, and secretes whatever's in there. That neurotransmitter then will diffuse across this synapse, just like we saw in muscle tissue function, which activates, again, chemically gated ion channels. You see this looks familiar, right? This is kind of the same thing we learned about in muscle physiology. So having said that, make sure you don't forget about how impulses are transmitted across a gap, and that these are chemically gated ion channels, and that they're going to kick in some voltage gated ion channels here in a minute. Now, here's a new term. This neurotransmitter, when it's released, if it's released from one neuron to another neuron, it's not going to generate an action potential immediately. If you remember that in muscle stimulation, the axon terminal would release neurotransmitter, and you'd immediately generate what we described as an action potential, where you basically change the polarization of the sarcolemma membrane, and that would cause change in polarization. It would cause this domino effect, which would knock down all these dominoes all the way down that muscle membrane, causing ultimately contraction. In this case, we're not generating action potentials, at least not immediately. Graded potentials have the ability to, they're a bit more subtle. So if you picture an action potential being like, if this is the membrane potential represented by where my hand is, an action potential is going to flip a switch and cause that potential to dramatically shift to the positive, and then you're going to have some other events which take over and reset the potential. But it's pretty much an all or none thing. You just turn the switch and the light comes on, turn the switch and an action potential generates. Graded potentials can either be excitatory or inhibitory, so they can either, and I've kind of drawn a little picture down here, an excitatory potential can be represented by, a depolarization of this membrane. Remember, if it starts off polarized and the membrane potential goes towards the positive, it's called depolarization. And that's likely to cause that neuron to send another impulse. So when I said earlier that neuron 1 is going to release some neurotransmitter, and it's going to either cause the neuron to do something or to not do something. Excitatory postsynaptic potentials, getting a lot of word salad here. Excitatory postsynaptic potentials increase the chance of the next neuron sending another impulse. So if that graded potential is positive, it's going to increase the likelihood that that second neuron is going to generate its own impulse. Now, when I said that they are graded, what I mean is that they don't have to go right to 11, right? You don't go straight up to the max every time. You can basically influence them to go in degrees, right? So that membrane potential can creep upwards, or based on other input, it can creep downwards. If it's creeping downwards, that's what's called an inhibitory postsynaptic potential, which I've written something similar over there, but whatever. Inhibitory postsynaptic potentials tend to decrease the likelihood of the postsynaptic neuron generating its own impulse. Now, keep in mind that these neurons, I'm going to draw my little neuron cell body again. So here's the crude version of it, right? These neurons are receiving input from all over the place. You could have dozens or hundreds or, who knows how many. Somebody knows. I don't know how many individual axon terminals can be interacting with this cell body at once. But you could have, 3/4 of them sending excitatory impulses and the other quarter sending inhibitory impulses. So it's basically kind of like a vote. If you generate enough excitatory potentials on this membrane of the cell body, you can get to this place right here, which you saw in the image, which you'll see in a minute again. You reach this area called an axon hillock. Sorry about the kind of compressed writing here. The axon hillock is basically where you tally up all the votes. So all of those impulses, all of those excitatory and inhibitory impulses are coming in from around this membrane. And when they get to this axon hillock, the axon hillock kind of sums it up and says, well, if it's above a certain level of excitatory influence, if it reaches a certain membrane potential, then the axon hillock is going to flip that switch that generates the action potential. And then we just pick up with a normal action potential as we learned in the last chapter. But that's why your brain doesn't have, it's not fire or don't fire until you get to that to that axon. There's a lot of so-called, I could say decision making taking place. Now these neurons aren't thinking, but I guess electrical decision making. Wait right there. Let me see what I got. Yeah, let's go back over here. This is a good spot for this, I suppose. Just to point out that axon hillock again. There it is labeled. So it's kind of, a hillock's just a small hill. So the axon hillock is kind of like the little bump on the cell body that it decides if another action potential is being generated. Once this axon hillock flips that switch, it's going to fire an action potential all the way down. And those things are, as we'll see, kind of can go all the way down an axon without ever losing any power. Now I'm going up to kind of macroscopic here. And this is a nerve itself. So this right here, if you look at the smallest level here, this is an axon. So what we're dealing with right down here is a single neuron, a single fiber, axon fiber of a single nerve cell or neuron. These neurons are bundled into packages, and these packages are bundled into bigger packages. And you can see a lot of terminology that's really similar to what we dealt with in the muscle section, which is all you got to do is change out the suffix here, mycium, for neurium. And you've got the same organization with the same name. So if you remember epimycium, perimycium, endomycium with fascicles and fibers, you've got the same stuff here, except for just switch it out and say epineurium is the connective tissue. Again, it's a dense, irregular connective tissue layer around an entire nerve. So a nerve is about the same scale as your muscle was, a skeletal muscle. Now, that nerve has a bunch of blood vessels and connective tissue inside of it, and those surround these little bundles of neurons called fascicles. Same name as you found in the muscle cell, in the muscle. So a fascicle is a bundle of neurons or axons of neurons, and they're surrounded by a layer called the perineurium. So epineurium around the whole nerve, perineurium around one fascicle, and then the fascicle is again a bundle. By the way, that root fascicle, did I talk about this one? Fascicle is the same root as you get for fascism. Did you know that? So it's a bundle. So they kind of warped the meaning a little bit back there in the 30s. So this fascicle is a wrapper around a bunch of neuron axons. Mostly axons in here because these neurons extend outside, these nerves extend outside of your central nervous system, and they're kind of packaged for transport out to there. They branch and stuff and recombine with others, as we'll see in the next chapter. And then here you see those nice little, that myelin sheet, it says myelinated axon and Schwann cell around there. And I'll be talking about those guys in detail right next. All right, so this right now is what we call the neuroglia. So you might see neuroglia or neuroglial cells, and it's right up here with the central nervous system, peripheral nervous system ones on the left and central nervous system ones here on the right. There are far more types, there's several more, a couple more, twice as many types of central nervous system glial cell as peripheral. So let's talk about them first. Ependymal cells. Ependymal cells' main contribution is that they line these chambers in your brain called ventricles, and they secrete a substance called cerebrospinal fluid. And we'll talk more about cerebrospinal fluid in the next lecture, but kind of think about it like a buoyant solution for your brain and spinal cord that also provides sort of a stable ionic and thermal environment. So it's a real useful thing. It's like your brain's floating in a little thin bathtub its whole life. Microglia, these are phagocytes. So microglia are just basically a highly modified macrophage, which is itself a highly modified monocyte. So it's basically just a phagocytic cell that swims around around your brain, around your nervous tissue, and if you get an infection, they're there to help it, remove it. Astrocytes are interesting cells. Now they've got these little, looks like they've got extensions of little suckers all over them, right? Well, what these little suckers do is that they basically form a middleman between your blood. So one sucker, if I'm an astrocyte, I'm going to put couple of my limbs or whatever on a capillary, and I'm going to put the other limbs on neurons and on the brain cells, so that anything brought into my central nervous system by my blood has to go through security first, right? You can get, like, if I cut my hand on something and got bacteria introduced to my blood, there's a direct route from that cut to my brain. It goes down to my heart, out to my lungs, back to the heart, up to my brain. That's one pathway. So you can get pretty rapid introduction of pathogens to your central nervous system, which is why we have these guys present, right? So these guys kind of keep a distance between your blood and your brain. This is called your blood-brain barrier. Nice alliteration there. And it keeps, pathogenic things and toxic things out, and it does a lot. It's kind of like a support system for your neurons of your central nervous system. Lastly, we have oligodendrocytes, and oligodendrocytes are next to Schwann cells, probably not accidentally, because they both produce myelin. Myelin, as we'll see, dramatically accelerates the nerve's impulse, and I'll show you how it does that too. At least I'll try. Oligodendrocytes are the cells that produce the myelin in your central nervous system. Their cousins are called Schwann cells. We'll move out to the peripheral nervous system here. And Schwann cells have been more studied. That's why you get like chapters or at least many pages of text oftentimes on Schwann cells, but far fewer on oligodendrocytes, because it's hard to like study inside of the brain that with the person still thinking microscopically. Anyway, so these Schwann cells surround the axons. You can imagine an axon running through here, basically, and it runs down the tunnel like this. So, again, similar, secrete myelin, increase impulse transmission speed. And then lastly, satellite cells, and you can kind of think of satellite cells as peripheral astrocytes. They are going to surround cell bodies in ganglia, and there goes that term ganglia again. Ganglia are clusters of neuronal cell bodies found in the peripheral nervous system. You have them run down they'd be like all the way down with every spinal nerve that comes out. You're going to have a ganglion on each side, which is a cluster of these sensory cell bodies. So they need protection and regulation too. So satellite cells serve a similar purpose to astrocytes. See what I've got next here. Let's go down to, let me let's do this first. I'll just go here, and that way, maybe if I have to come back, I can. So here goes kind of a coming at you look at a neuron where you have your cell body, and we'll just assume that there was enough excitatory graded potentials generated here to get the axon hillock to send an action potential. Now, these hot dog buns are, you might hear them called insulators. They insulate the axon. They're not insulating it to keep it warm. They're insulating it like the rubber around an electrical wire insulates it. So it's going to restrict the flow of electricity in and out of that wire. In this case, we're talking about ions. We're talking about sodium and potassium, the same ions we dealt with in the muscular system. So they're going to keep the ions from going into or out of this axon, except for at these key points. So you may depolarize this region right here. The axon hillock, in fact, does depolarize. And when it depolarizes, this is something, here's my disclaimer on this. You remember in the muscle tissue chapter from chapter 10, when I said, here's how an action potential works, and sodium goes in, depolarization, potassium goes out, repolarization, hyperpolarization, sodium potassium pump, all that. I'm not going to say it again, but go back and study that part, because I'm going to ask you questions about the action potentials on this material. But note to self, go back to chapter 10 video and study action potential again. So an action potential is generated here, but now it's kind of like trapped. It can't go, it can't travel in a wave. You can't have sodium that's located out here move inside like this, because this myelin here is surrounding this axon. So what happens is, if I get a depletion of sodium in this area, because it's all gone inside, and I get a depletion of potassium in here because it's all gone outside, basically that's what depolarization is, but it's going to draw sodium in from the surrounding areas, and it's going to draw potassium in from the surrounding areas, which is going to act to depolarize the next little spot. So the depolarization doesn't travel in a wave, which as it turns out takes longer. It travels in these jumps, and they draw arrows like it's wee jumping over, but it's not. It's just boom, it kind of like teleports. It depolarizes here, and then that changes that electromagnetic field enough that it's going to depolarize here. And then it's going to depolarize here. It's like it's going to trip each switch like a millimeter down the road. And that may not seem like a lot, but a millimeter is a pretty good distance in cell terms, right? You have to travel them all the way down your arm or whatever, but it goes much faster that way. This type of impulse transmission is called saltatory conduction, and I have it written on the board, but saltation or saltatory locomotion, interestingly enough, maybe, is how kangaroos and rabbits move. So if you ever want to sound like a real nerd and you see something hopping along, say, it seems to be exhibiting saltatory locomotion. And wait for the applause or people to walk away. All right, so in any case, let's go back over to the board over here where I talk about the myelin sheath specifically and tell you some deets about it. So it is a protein lipoid, and fats tend to act as electrical insulators. So this is a protein with lipid functional groups, and it does just that. It's an electrical insulator. The gaps in between those things are called nodes of Ranvier. So it looks like Ranvier, but it's French, so it's Ranvier. So this is a node of Ranvier, this is a node of Ranvier, and here's the little wee saltatory conduction, which just means jumping conduction of electricity. Here I've shown you an unmyelinated axon with the flipping of that action potential switch traveling in a very wave-like motion, whereas down here, because it can't depolarize and repolarize within this area of this myelin sheath, basically I've drawn the sodium and potassium leaving the area, depolarizing this, and so on. What else? Okay, now let's just a little refresher, brief refresher course on action potentials just to compare them with graded potentials. Action potentials are old hat. They're basically a binary. So if you reach that impulse threshold, if you depolarize that membrane enough, you get an action potential generated. Action potentials travel down axons. We've already established that. It acts in a way similar to muscles. This line right here is what I was preaching at you over there. Go back to that chapter and look at your action potential generation graphs and data. Action potentials are self-propagating, which means that they will cause an action potential causes another action potential, which causes another action potential, so it acts just like dominoes, where if you have, I could push, you ever see those videos where they'll push over a tiny domino, which will knock over a slightly bigger domino, which will knock over a slightly bigger domino. You can knock over a building like that, theoretically. But you can certainly knock over something the same size. So the signal doesn't degrade over time. When I generate an action potential, it just knocks down the depolarizations all the way down that axon or all the way down that sarcolemma. Contrast that with the graded potentials. Graded potentials are graded, meaning that they don't go all or none. They can creep up or creep down. They are found being they transmit along dendrites in the cell body, so that the receiving portions of the neuron generate graded potentials. They are decremental, which means that they degrade over time. If you've ever taken a rope, you ever do that rope workout thing? You ever do that where you just go like this, right? And you're whipping those ropes? So, but just picture one rope. If you just took one rope and whoosh, whipped it, and it's attached or whatever at the end, you'll find that you create like a wave in that rope. Well, that wave, as it travels toward the other end of the room, doesn't stay the same height, right? It's going to lose amplitude as you go. So when I whip that rope, it's going to go and kind of peter out over time. That's how you can think of a graded potential. That you can whip it, but if you don't whip it hard enough, and if you don't whip it close enough to the axon hillock, it's going to peter out over time. So they don't have a lot of range. This is why you oftentimes get lots of axon terminals stimulating the same neuron to get a real big likelihood of a subsequent impulse generation. They can be excitatory, inhibitory. That's what I mentioned earlier. And I've kind of modified what an action potential looks like. This would be representative of an action potential. Actually, it doesn't have anything to do with excitatory or inhibitory. This graph has to do with self-propagating and decremental. This one, being self-propagating, represents an action potential. And this one over here represents a graded potential. So if I kick that depolarization up, it's going to kick the next one up, which is going to kick the next one up. So you can see that action potentials retain their amplitude the whole way. Graded potentials degrade over time. So that first depolarization event results in a smaller depolarization event, which results in a smaller yet smaller depolarization event, which then can ultimately lead to basically no change in membrane potential. Now you may think this is just off script here, but you might think, well, why do I do that? Why don't I just say generate an impulse, go? Well, you don't want your brain cells responding like a reflex to every impulse, right? When you go to the doctor and they go boink and your knee kicks, or if you put your hand on a hot stove and you jerk your hand back, that's an automatic response. You don't have to think about it. It's probably in your best interest. I'll tell you about the knee one later. But when you're processing myriad information in your brain from many different sources, you don't want to jump to you don't want to jump to a neurological conclusion by just one impulse comes in, send off a bunch of impulses, right? You want to basically weigh your data here. You want to kind of take a good look at what the situation is before you speak or step or act or whatever. That's why your brain does best when it is a democracy rather than a autocracy. All right, I think I'm on the last thing here. And that is this last little frame over here. Do you got it? Can you see it? I'll move over to this side. That doesn't matter. So this has to do with conduction velocities and fiber type. So I should probably draw a line in there, but whatever. You see we've got 1, 2, 3. There's more, but I'm just simplifying it. There's 3 basic fiber types. Maybe we'll go back over here. So am I screwing you up? Screwing up the cinematography here. So group A fibers are, I don't know if they're your most common, but that's kind of, I think I remember that. Let's not etch that in stone. Group A fibers, what they look like is they're very large in diameter. So these are larger fibers. And whenever you hear the word fiber, think axon. So fiber is axon, which is part of a neuron, just like fiber in the muscular system was cell. So these are large diameter axons with a lot of myelination. I've tried to draw what that might look like. You know, very uniform, not big gaps between the myelin sheath units. By the way, the Schwann cells are what are wrapped around there creating that myelin. Their functions or their purposes are sensory and motor to your skin, muscles, and your joints, right? So, and then the impulse speeds are really great. This is up to 150 meters per second. I'll give you some perspective on that here in a second. But we want big, fast fibers going to our skin and our muscles and to our articulations. Why is that? Well, because when you're out there, you can stumble off of a cliff real easily if you're not careful. You can trip on a stone, right? You guys have all stepped off of a curb or whatever, right? You've gone and you walk, right? Well, when you do that, you don't just want to go, you don't want to twist your ankle or whatever, or make a misstep and just, just fall every time. You want to be able to maybe recover yourself because falling down can hurt you, right? Or if an aggressive person comes at you, or a bear, right? If a bear's coming at you, you want to be able to run as fast as you can past your slow friend, so that when the bear jumps out, you find the slowest person in your camp and you run past him or her, because the bear then will catch them. But you're fast, right? So you're going to beat them. But you want big fibers, you want big fast fibers going there so that you have the fast reaction times. When you catch something, right? It looks easy. Walking looks easy, right? When I walk, let me give you a little story here. When you walk, you're basically pole vaulting off of 1 foot. I don't know how much it's in frame here, but you're going to pole vault off of 1 foot. All right, yeah, we got some in film script changes. So you're going to pole vault off of 1 foot, and then I'm up in the air, tilting forward, and I'm going to fall forward onto the next foot. And then I'm going to vault up onto that foot, and I'm going to fall forward onto the next foot, and so on, right? Every step, I need to have instantaneous feedback from where that foot is, and where my center of gravity is, and then my brain's got to process all that, send out impulses to all my muscles, which then have to adapt. It may not seem like you're doing much, but you may have to take a shorter step or a longer step. And all this stuff can be done very easily because we've got these fast fibers. When you get certain disorders that can degrade the myelination, stuff like multiple sclerosis, which ultimately results in degradation of the myelin sheath on these grade A, class A fibers, those individuals end up having significant difficulty with tasks that we take for granted. So if the impulse transmission is not coming at 150 meters per second, by the way, if you know how fast that is, that's, if you've ever run track, that's 1 straightaway and half of a curve in a second. And so that would be, the fastest people on Earth now could probably do that in about 15 seconds. So it's 10 times faster than the fastest person on Earth can run. It's like about 300 miles an hour. If you have slower transmission rates, either sensory or motor, you're going to have trouble. And I've had friends with multiple sclerosis where as that progresses, they have to make sure that they get where they're going next, and they have to basically put their center of gravity over those 2 feet, move their body forward, because they're not going to respond well to an accidental trip or a misstep. That's just going to take you out. This costs a lot of energy. It costs a lot of energy to build big fibers and to make a lot of myelin. It's not free metabolically. So if you don't need the rapid response times, you can build smaller fibers with less myelin. And these are group B and C fibers. They're similar. Group B just is less myelinated. So I've kind of tried to draw that there. And they tend to be somewhat thinner. So somewhere in the middle on diameter and myelination. These are going to be autonomic functions like impulses if you're going to have to sweat, or your heart rate's going to go up, or your vasodilation or vasoconstriction, things that don't need to respond in a fraction of a second, right? If you're a little hot, you can wait a second before you start sweating. And this is about 15 meters per second. 15 meters per second would be, pretty fast. That's still fast, but it's 1/10 as fast as the group A fibers. So these go to your stomach. They go to your organs. If you're full, you've got sensory fibers. They're going to send it up, either group B or group C fibers, telling you you're getting full. You don't need that. You're not going to, stop that next bite. And then group C, they're the smallest. They're non-myelinated. They have a similar function to group B, and they only move 1 meter per second about. So 1 meter per second is like this fast. If I walk off screen. So like a slow walk. You definitely don't want your impulses from your muscles and joints and skin coming back to you at a slow walk. By the way, all that myelination occurs in your brain as well. You'll see when we get to the gross anatomy of the nervous system that you've got stuff called white matter and gray matter. White matter is white because it's loaded with myelin. So your brain's thinking real fast in order to get that information out to your body as well. That's it. It's somewhat truncated. You're welcome. Chapter 11, the first chapter of the last unit. See you later.